Okay, so I'm going to talk about how to boost the credibility of your research. And specifically, I'm going to talk about resources that can actually help you do that. And I am very grateful that the EDA team was awarded the Credibility Prize this year. So the EDA is obviously one of the resources that I will cover. So I'm from the National Center for the 3Rs, and we've got an entire program of work that's dedicated to supporting researchers with the design and the reporting of their experiments. And that's to ensure that experiments are as robust and reliable as possible so that they can truly add to the knowledge base. So my talk is going to focus on animal and preclinical research because this is the type of research that is within NC3Rs remit. So I suspect you're already familiar with the issue of reproducibility in animal research. Antonina provided useful background earlier in her talk. And this is a topic that has been discussed at length. So many scientists and organizations are concerned about the quality of animal experiments. These are just a few of the headlines from the past few years. It's actually estimated that less than 50% of preclinical research is reproducible. So if you apply this estimation to research done in the UK, this could mean a waste of 1.6 million animals every year. So improving the standards is important from an ethical perspective, but there are also wider implications for the development of therapeutics. So irreproducible findings published in the scientific literature can trigger clinical studies and subject patients to treatment that are unlikely to be effective. And it can also delay the discovery of new treatments. So improving the rigor and the transparency of animal research will improve the entire drug development process. And there are many factors that contribute to this issue. And the main ones are a lack of rigor in the design and the analysis of the experiments, and incomplete reporting in scientific publications. So in order to help scientists design robust animal experiments, we developed a great tool called the Experimental Design Assistant, or EDA for friends. It's an online system which is freely available. You can design your experiment in the system, and it's like your own personal statistical assistant. It provides bespoke advice and feedback on your experimental and statistical plans. So that also includes dedicated tools for randomization, blinding, and power calculation. So you can do all of that from inside the system. And basically, it helps you design rigorous animal experiments so that you can get reliable and reproducible results. So in the EDA, each experimental plan is represented as a diagram with high-level information about the experiment and the objective, the practical steps carried out in the lab, and the analysis plan. And it's a bit like Lego bricks. You can combine them in any way that you want. You can use as many bricks as you want to represent the experiment that you want to do. And the diagrams are machine readable. So the system uses computer-based logical reasoning to provide feedback and advice. So for example, this can be the type of feedback that you get, warning you that nuisance variables have not been identified for this experiment. So the prompt will include what a nuisance variable is, the common type of nuisance variables in animal experiments, like baseline body weight or the day of the experiment if there's day-to-day variability, and how to account for the variability in the design and the analysis of your experiment. And this is another type of feedback. This is a recommendation for a method of analysis that's compatible with the design that the experiment you've just designed. So in this case, this would be an ANOVA with blocking factors. So the system will analyze your diagram and provide you with feedback to help you make the design more rigorous. And the EDA took several years to develop. It's actually still in continuous development, and we keep on adding new features and new improvements. And there's a massive team behind it. I don't have everyone pictures, sorry, but this was developed as a collaboration between the NC3Rs, Certus Technologies, who specializes in innovative software for the life sciences, and a working group of experts in experimental design and in vivo research, both from academia and industry. So when we started working together in 2011, we really saw a gap in the market in that there were books or courses available on experimental design and statistics, but scientists are busy people. They don't have time to take time away from the research to become experts in statistics. And also, many basic scientists do not have the support of a statistician to help them design experiments, let alone get statistical advice tailored to animal experiments. And that tailoring is really important because, for example, the sources of variability or the sources of bias are going to be different in an animal experiment compared to a clinical trial. So a clinical statistician or an epidemiologist is not going to ask you the relevant questions if they're not familiar with animal research. So we wanted to develop a system where you can learn about design and statistics while working on your own experiment, without taking time away from your research. And the EDA does that. Every step of the way, it provides you with the specific information that you need to make an informed decision. It will not design the experiment for you, but it will give you just enough information so that you can design it rigorously and you can produce credible research. And another important tool in the toolbox to improve reproducibility are reporting guidelines. I'm sure many of you will have heard about the RAV guidelines. These are guidelines of a checklist of key information to include in the scientific publication so that the study is described in a comprehensive manner. And the research can be evaluated adequately and the methods and findings can be reproduced. So they were originally published in 2010. They've been recently updated. The new version was released in summer 2020. And these guidelines really apply to all in vivo research, whether using a mammalian species or an invertebrate. It doesn't matter if the species used does not require ethical approval. The guidelines are about rigorous science, so that applies to any experiment. And since the update in 2020, they're now split in two sets. The essential 10, which are the basic minimum to include in the manuscript, because without this information, readers and reviewers cannot assess the reliability of the findings, and the recommended set, which complements the essential 10 and add important context to the study. So reporting the items in both sets represent best practice. And when we updated the guidelines, we also developed an explanation and elaboration document, which was published alongside in PLOS Biology, and it's also available from their RAV website. And this explains the rationale behind each item of the guideline and why it's important to report this information. It also clarifies key concepts, particularly around experimental data and statistics. And it provides examples from the published literature showing how to report this information. And we will gradually increase the number of examples for each item of the guidelines so that we can provide examples of good reporting in a wide range of model organisms and experimental settings. So that document in the RAV website is definitely something that I recommend that you consult before even starting designing your experiments, because all of this concept, all of the statistical concepts are going to be really useful, and it's going to be very useful for you to know, what information you're going to have to report before you start, so that you can collect this information. You can make sure that you design the experiment in a way that you'd be happy to report it in a published paper. So, and we are currently working on recommendations to improve the reporting of in vitro research, which will be released by the end of the year, so watch this space. Okay, so that's my take-home message. One thing to boost the credibility of your animal experiments today, I would say register for an EDK account. It takes 2 minutes to register, and from there you can have a play around. You can try building a diagram representing your next animal experiment, and you can get a feel for the system. So you can just log on onto the EDA there. The URL is on the screen. And that's it. Thank you very much for your attention. I'm happy to take any question. I've added further links on that slides for your reference. And I'm also happy to take the discussion to Padlet later as well.
Thanks, Nathalie. And yeah, congratulations again on what looks to be a fantastic tool. We have a question already. The EDA tool sounds extremely useful. Do you think it would be used for human experiments or non-clinical trials?
Yes, so that's a question that's been asked loads and loads and loads of time, because when I present the tool, people get very jealous when they do human research because they want to use it as well. So the principle are the same. So if you really wanted to, plan a human experiment in the tool, you could, just consider that humans are animals, basically. But I think where the advice differs is that, as I mentioned, the tailoring to animal research is very important. So a lot of the feedback that the EDK is going to provide is actually based on an animal experiment, on what you're expected to see in an animal experiment, on the problems that you might come across, on the fact that you've got multiple outcome measures often. So it's that kind of thing. The EDA is very much geared towards giving you that specific advice that will be relevant to animal experiment. And in this, that advice might not be relevant to human research, like, where the cages on the rack are, is not going to be relevant to human research. But I know of a few teams that are actually looking into adapting the EDA to human research, but that would mean creating, duplicating the EDA and then drafting advice that would be relevant to human research. And we're also planning to do that for in vitro research. So this is kind of like, not in development yet, but we will start working on that soon at the NC3Rs. so that there can be a tool that can help the design of in vitro research.
Thank you. Another question here, or a tough one. Are the recommendations in ARRIVE based on experimental evidence?
Yes, a lot of them are evidence-based, obviously. So if you think of the essential 10 in ARRIVE guidelines, it's all based on good experimental design, and we've got ample evidence showing that randomization, blinding, exclusion criteria, and such will actually have an influence on the outcome and the effect sizes. So that's why it's really, really important to include this information in your publication. Even if you said that you've not done it, no experiment is perfect. Animal experiments are really, really difficult. You're always making concessions, you're making hard choices. Sometimes it's not possible to blind because you're the only one working on this experiment and you can't just blind on your own. Sometimes the blinding would actually risk making a massive mistake and then losing your entire data set. So sometimes it is not possible, but I think what we need is transparency so that we can actually, and also with that transparency, we'll be able to continue assessing how big the effect is on blinding, on randomization, on having a priori exclusion criteria. So I think we've done as much as we could based on what's currently published. But if people started being more and more transparent, then we'd be able to assess this more precisely. But that's definitely based on evidence.
Thank you. And Lan asks, would EDA be eventually acquired by all funding bodies? I noticed that some ask for EDA, e.g.
Yes, so yeah, many major funding bodies recommend that researchers use the EDA when preparing the grant application. And that's because when you use the EDA, you're actually going to be thinking about your design and you're going to be identifying the information that you're going to have to put in your grant application anyway. For example, for MRC grant or NC3Rs grant, you've got a section on experimental design that you've got to fill in. So all this information can be identified with the EDA. The EDA can actually help you identify this information. There's also more direct ways. I mean, from the EDA, you can actually generate reports that you can stick into your grant application directly. So that's not accepted for all schemes and not all funders. So I would recommend that you actually check for your specific scheme and the specific funder you're applying for. But that's definitely, yeah, I mean, funders definitely recommend use of the EDA. They can see the benefit of it. Funders want to fund, high quality research. They want to fund rigorous research. So they do recommend using that tool. And that's not just in the UK, that's worldwide. And the NIH recommends that researchers use the EDA for the funding application as well.
Thank you. Catherine asks, a lot of genetics research on animals, including ours, uses littermate controls rather than randomization. Is this justifiable? It does reduce numbers.
So I'm not going to comment on the reduction in numbers because I'm not familiar with that type of research enough to comment on this. But in terms of I think it's a hard question. There's several levels in there. So using littermate, definitely, there's obviously no problem with that. I think you actually really need to think about the design and then decide whether the experimental unit is the entire litter or whether that's an animal within a litter. And then that will actually send you towards different design type. And if the animal is the experimental unit, then that's the thing that needs to be randomized, but the litter can be a blocking factor. So there's lots of different things that you need to think about the design, but this is definitely a valid approach. And there are ways that you could randomize within that approach.
I don't see any more questions, so I'll just ask final one myself. So once you've designed your experiment with EDA, presumably it can produce a machine-readable description of that experiment, which you could then upload to some kind of pre-registration website to lock and make publicly available so that you're perfectly transparent in what you plan to do before you do it. So is that a plan? I mean, because current pre-registrations on the websites are still verbal descriptions, which still have a few degrees of freedom and ambiguity about them, whereas a machine-readable specification is pretty hard to argue with. Have people done that? Would you want to do that? Is that easy to do?
Yeah, I mean, we actually we were in discussion with the Center for Open Science to have a way of actually just directly exporting an EDA output into a pre-registration.
Great.
So that's definitely something that would be valuable, because it would also save a lot of time. Like, if you actually design your experiment in EDA, and you can export it at the click of a button rather than having to do an entire new pre-registration somewhere. I think that's really valuable for researchers as well. And the information content in the EDA diagram is so much more than a text description. So yes, that's something that we are looking into. We are also looking into having an EDA output that you can include in your publication. So obviously in your manuscript, you're going to include, your methods and your results and so on. But having just, the EDA diagram with it will provide so much more information. You still need to include enough information in your publication so that readers can actually understand what you've done. But the EDA will actually provide put it to the next level and also clarify what things are. I mean, with an EDA diagram, the design is very obvious. It's very obvious how many groups you have, how many treatments you're giving, and so on, because they're all visually represented. And then you've got all this things that are much harder to talk about, like the variables and what analysis the variables were included in. So having that visual representation really clarifies whatever description you might have put in your paper already.
I agree. As a reviewer of many papers, it's often unclear for the meta section exactly what statistics they've done, and it's much better to share code or machine readable